So this is Rekha from Government Science College, Bangalore. So today we shall discuss the mechanism of translocation of organic solutes. So there are several theories to explain the mechanism of solute translocation. It is a hot debated problem in physiology. The problem may be, however, been visualized as short distance translocation and long distance translocation. Of the several theories proposed, short distance translocation may be explained by protoplasmic streaming theory, transcellular streaming theory, contractile proteins, and electroosmosis. The long distance translocation is explained by pressure flow theory, mass flow theory or Munch hypothesis, and bimodal theory. So today we shall restrict only to the short distance translocation theories, that is protoplasmic streaming, transcellular streaming hypothesis, and contractive protein hypothesis. So first one is protoplasmic streaming theory. This theory was proposed by Hugo de Vries in 1885 based on the observation of protoplasmic streaming in the sieve elements. So according to this theory, the movement of the solute solution is caused by a combination of diffusion and cytoplasmic streaming. The diffusion would occur from sieve tube to a sieve tube across the intervening cross walls through the pores, and within a sieve tube a substance would be carried downward or upward as the case may be by the cytoplasmic streaming. So thus the solute translocation is a combination of diffusion and protoplasmic streaming. So the second one is transcellular streaming. So this theory was proposed by Thine et al. In the year 1964. So according to this theory, the sieve tubes have a number of cytoplasmic strands called the transcellular strands. So these transcellular strands are proteinaceous and they have microtubules helping in the rhythmic contraction. The organic solutes are assumed to move along these strands from one sieve tube element to another sieve tube element. So the different strands which are present in a single sieve tube would be able to move sugar in opposite directions at the same time. So this explains the bidirectional flow of solutes. And this is the diagram, the LS of sieve tube, which shows the transcellular strands, and it also the second diagram is the cross section of the same sieve tube. So objections of this theory includes how this biological energy could be transduced into a physical force within the sieve tube. The nature of the transcellular strands, are they solid or tubular? Whether the solute particle move in or on the strands, and whether the strands are not seen when sieve tubes are observed under electron microscopes. So these were the major objections of transcellular streaming theory. The third one is contractile protein theory. So this was proposed by Fensham and Williams in the year 1974, and they reported the presence of a network of microfibrils in the sieve tubes. These microfibrils oscillate resembling the moving flagella and bring about the solute migration within a sieve tube. The microfibrils are composed of contractile threads of P protein. So coming to the summary of this short distance translocation theories, many of the evidences favor the translocation of organic solutes through phloem, but however, no unanimity regarding the mechanism of nutrient flow. And there are a number of views and hypotheses proposed by different scientists to explain the mechanism for short distance as well as long distance translocation. It is quite possible that nutrient flow in phloem is driven by more than one mechanism, or different mechanism might operate in different plants or in different tissues of the same plant. Thank you for listening my lecture.